In Unit 3.2, we'll be focusing on neural communication. Remember, as we discussed in class, the main focus here are parts and functions. That's right. So as we explore the dendrite and its many parts and how it communicates, remember to know its parts and its functions of those many parts. So while Franz Gall was on the right track with his theory that specific parts of the brain have various and specific functions, however, he was wrong in his hypothesis that the shape of the skull and physical features dictate innate personality traits. It is actually much more complex than that. It is biochemical, it is genetics, it is neurological phenomenon that direct and guide our behavior, not outward physical appearance. This is what we will begin to explore for the rest of this unit. We begin our exploration of the biology of behavior with first the neuron. The neuron, or a neuron, is the nerve cell that is the basic building block to our entire nervous system. All right, you can find neurons in the brain, you can find neurons branching all throughout your body, and you can also find neurons inside your spinal cord. All right, we're going to be exploring all these many neurons, but before we do, we are going to be highlighting the specific parts of an individual neuron so that we can better understand how neurons communicate with one another and how our physiological and biochemical processes are able to be processed almost simultaneously. It happens so fast due to electrochemical signals that we can't even really detect how quickly we process information. Okay. It's also important to note that our body is comprised of over 100 billion interconnected neurons, and some of these neurons can be different shapes and different sizes. For example, there's one neuron that stretches the length of our leg, whereas other neurons are microscopic. You can't even see them. All right, so we're going to be discussing really just the basic parts of a neuron and what those parts do before we start going in more depth and talking about neural communication. So as we see here, this is an image of a neuron, okay, and all its many parts. All right, we're going to first start with the four of the basic parts of the neuron, the first being the dendrites. Now, the dendrites are these branching areas of the neuron here. And what the purpose of the dendrites, which looks like a branch on a tree, they carry information to the cell of the neuron. So the dendrite absorbs incoming information. They absorb neurotransmitters. All right, so the signal will go into the dendrite. The cell body, also known as the soma, is the larger gray area of the neuron, as you can see here. And the soma contains the nucleus. And the nucleus is where the threshold is located. And we're going to talk more about that later. All right, the nucleus or the soma is also where all the nutrition for the neuron is stored. And this is what helps to develop and facilitate a healthy functioning neuron. The axon is what carries information to the next cell. The axon is this very long, almost snake-like structure of the neuron. Okay, this is where action potential takes place, and we'll talk more about that later. The myelin sheath are these yellow little pod-looking things that wrap around the axon, as you can see here in this image. All right, the purpose of the myelin sheath, first of all, it's a glial cell. It's a type of cell called a glial cell. And the purpose of the myelin sheath is that it insulates the axon and it speeds up action potential, or it speeds up neural impulses. Okay? So how does it insulate? How does it protect the neuron? Because neurons are so interconnected and they're so densely compacted against one another, the myelin sheath helps to protect one neuron from the electrochemical processes of the next neuron, so that each neuron can function independently from another and without causing harm to another. So once again, here we have the myelin sheath, the neuron. Okay. Now remember, the neurotransmitters will be absorbed by the dendrites, which are these branching extensions, and then that electrochemical signal is then passed to the soma, also known as the cell body. In which case, if the threshold of the soma is surpassed, then action potential will ensue down the axon, which is accelerated by the myelin sheath, as we talked about, and then it releases neurotransmitters from the terminal branches, also known as the end bulbs, and then they will go on to the next neuron, which will then be absorbed by the dendrites, and the whole process will repeat all over again. Here we see a large neuron with some of its parts labeled. You can see other smaller neurons. Notice all the small black-looking fibers. Those are called terminal branches, and they branch out from other neurons to form a very tightly woven web of neurons. Millions and millions of these tightly woven webs of neurons can form nerves that branch throughout the body, and they can also weave together to form densely connected neural connections in the brain. While there are different types of odd neurons that come in all different shapes and sizes, we will initially focus on the basic parts of a single neuron and build from there. So first, I'm going to go through some more images of the neuron and explain its many parts and functions of the neuron, as well as some other interesting information. But for those of you who want the concepts and definitions that you need to know for the test, be patient, they are coming. So again, we start with the dendrites. And remember, dendrites receive incoming signals. They receive neurotransmitters. And then after that signal is accepted by the dendrites, it's passed to the soma or cell body, which maintains the health of the neuron, as we've already discussed. And then it's passed down the axon. All right, this passing through the axon is known as a neural impulse, which is electrical signals traveling down the axon. And then it's accelerated by the myelin sheath, all right? And then it reaches the terminal branches, which are these, releasing neurotransmitters from the end bulbs on the terminal branches. These little buttons are called end bulbs, or terminal buttons. They have a lot of different names. We'll talk about that in class. I like this image because it shows how the neurons are interconnected. Now, they're not as densely interconnected in this picture as they are in reality, but you can see them how they somewhat connect there. Another reason I love this image is because you can see that the myelin sheath, which is, again, a glial cell, it's wrapped around the axon. All right, this is how it's like in reality because it provides insulation and it also accelerates action potential. Here's another image of the neuron, and it also shows the direction of action potential. As you see the arrows pointing, you might want to take a second, pause, and just analyze this image and just think about it for a second. Here's an image of an actual neuron. The scientists who took this image actually used different colored dyes, and that's how you can see the different parts of the neuron. All right, so it doesn't look exactly like the diagram and the illustration that we've been looking at, but it still has those main parts and functions that you need to know. And here's a cluster of neurons. It's hard to see an individual neuron because they're so interconnected and it's so dense. But this is a cluster of neurons that was taken a picture of in the occipital lobe, the visual cortex of the brain, where we process visual sensory information. This image shows is an image taken by an electron micrograph. And here you can see the end bulbs, or also known as the axon terminals. Here, these are the many little like inner tube looking things, and they are connecting to a dendrite, which is the larger thing here, and in between them is called the synapse, and this is where the neurotransmitters will be exchanged and which facilitates neural communication. So we've talked about the myelin sheath, we've talked about how it's a glial cell. Well, just real quickly, let's talk about a glial cell. Glial cells are cells that insulate and support neurons. They create, the glial cell creates the myelin sheath. It removes waste products, provides nourishment, and prevents harmful substances from entering the brain. There are many different types of glial cells, but the one that we need to be concerned with in this class is the myelin sheath. Okay, so you need to know what the purpose of a glial cell is, and then specifically what the myelin sheath does for the neuron. That is, it insulates it, provides nourishment, and accelerates action potential. So once again, in review, dendrites are these tree branches that come out of the neuron, come off, that extend from the neuron, and what they do is they absorb or they receive neurotransmitters, and then that message from the neurotransmitter is then passed on to the soma or cell body, where the threshold of the nucleus is either surpassed or not. Once again, the soma, also known as the cell body, is the home of the nucleus. This stores most of the nutrients of the neuron. The nucleus is within the soma, determines the occurrence of action potential. So whether the neuron fires or not, that is up to the nucleus. And this is called the threshold, as we see here. All right. The threshold, if it's surpassed, then action potential takes place, and an electrochemical impulse is surged through the axon, releasing neurotransmitters from the end bulbs of the terminal branches, and then those neurotransmitters go into the dendrite of the next neuron. All right. That is called the threshold in the nucleus. Here. Okay. The axon, again, is the long extension of the neuron. It's the long fiber ending in the branching terminals, as you see here, through which messages are sent to other neurons or to muscles or other glands. At the end of the axon are thousands of terminal buttons that connect to either muscles, glands, or other adjacent neurons. As we said, the myelin sheath accelerates action potential. It also provides nourishment to the axon. For those people who suffer from multiple sclerosis, the cause for multiple sclerosis is a degeneration of the myelin sheath. It's because the myelin sheath in their neurons are deficient, so that they are unable to provide as much nourishment. They're unable to accelerate action potential like a normal neuron, a normal functioning neuron would be able to do. And so for that reason, they will have multiple sclerosis. As you can see, there are little areas between the myelin sheath. These little spaces between the myelin sheath are known as the nodes of Ranvier. And this is the space on the axon between the myelin sheath where electrical impulses are generated that speed up action potential. And again, the terminal buttons, these are the end bulbs, also known as presynaptic cleft, also known as the axon terminals. There's a lot of names for them. You need to be familiar with all of them. These form the junctions with other cells or neurons, and they house the vesicles, which also in turn house the neurotransmitters, which are released after action potential. So I just mentioned vesicles. What are those? All right, so here we see the end bulbs that we've been talking about, and they're making connections with the dendrite of the yellow neuron. Okay, and so here's a closer look as to what that actually looks like. You see the balloons, the little bubbles there. You can see it better here. There's a bubble that has a red dot on the inside. Well, that larger bubble, that's the vesicle. And the vesicle is housed in the end bulb of the axon terminal. And inside those vesicles are little neurotransmitters. And these neurotransmitters are what carry the signal and communicate what we're trying to do, whether we're moving a muscle, we're trying to memorize a piece of music. These neurotransmitters, depending on which one they are, that is what facilitates that sharing of information in the neural communication. All right, so the vesicles are popped after action potential occurs, releasing the neurotransmitters to the adjacent neuron. Here's a real-life electromagnetic image of that. Here we see the vesicles. The small vesicles here, you can't see the neurons within them because it's too small. And this area, the space in between the presynaptic terminal and the dendrite is known as the synapse. Here we see it, the synaptic cleft. We're going to be discussing that shortly. So by the end of this unit, 3.2, you should be able to explain this chart and more. You already know the basic parts of the neuron and their functions, but now we will learn the really important part, how one neuron communicates with another, and then another, and then another 1,000,000 times over. While neural communication involves a lot of functions from a lot of parts of the neuron, it takes place at one location and one location only, the synapse. And that is where we are going to begin our exploration of neural communication. As we said two slides ago, the synapse, or the synaptic cleft, or synaptic gap, is the space between the end bulb of the axon terminal and the dendrite of the next neuron. All right? So it's the space between the axon tip and of the sending neuron and the dendrite of the receiving neuron. Neurons work by electricity. They are electrochemical responses, which will not go over a space, so it stops. I love this image because it shows us pretty much everything we need to know at this point. All right, so here we see the dendrites, there's the soma, there's the nucleus, and then when action potential occurs, it travels down the axon, accelerated by the myelin sheath. It reaches the axon terminals. It then reaches the end bulbs or terminal buttons, which then pop the vesicles that are housed in the terminal buttons, which then release the neurotransmitters across the synapse and into the next neuron, the dendrite of the next neuron, as we see here. Okay, so what are these neurotransmitters that I've been talking about? These neurotransmitters are key. They are the main ingredient to neural communication and enable us to do everything that we are able to do as human beings. All right, so neurotransmitters are first chemical messengers that traverse the synaptic gaps between neurons. When released by the sending neuron, neurotransmitters travel across the synapse and bind to the receptor sites on the receiving neuron, thereby influencing whether it will generate a neural impulse. If the message is for arm movement, the vesicles only release neurotransmitters involved in the movement circuit. There are dozens of different neurotransmitters, and we're going to learn about 7 today. But before we do, let's go over some parts. Remember the synaptic vesicles. These are the vesicles that are housed in the end bulbs, also known as the presynaptic cleft. They are sacs and terminal buttons that release chemicals into the synaptic space. They hold the neurotransmitters. Again, neurotransmitters being the chemicals released by the synaptic vesicles. And then the receptor sites on the dendrite of the next neuron are locations where the neuron receives specific neurotransmitters. This is a chart on table 2.1. It's on page 58 in your textbook. You need to memorize this chart because it shows you the parts of the neurotransmitters, the functions of the neurotransmitters, and examples of what happens if those neurotransmitters malfunction, which you need to be knowledgeable on all of these because you could see it on the test and the AP exam. So the first neurotransmitter we're going to talk about is known as ACH, also known as acetylcholine. It is the most common and best understood neurotransmitter. This neurotransmitter allows for muscle movements and it also facilitates memory. All right. So a lack of acetylcholine can lead to Alzheimer's disease, which is the loss of memory. The next neurotransmitter are known as endorphins. All right, these are known as the morphine within. What these do, it's a natural opiate essentially, so it makes you feel good. It gives you this sense of well-being and relaxation. Dopamine is a neurotransmitter that's involved in the control of bodily movements, learning, attention, and emotion. Shortage causes Parkinson's disease, and excessive dopamine is linked with schizophrenia. All right. Now, we said earlier that ACH is used in body movements, so what's the difference between ACH and dopamine in terms of bodily movements? Well, ACH is more muscle contractions, so if you can imagine someone doing the robot, like the dance, that's muscle contractions, whereas dopamine is more fluid body movements, so like someone doing the wave, for example, or the worm, okay? Serotonin is the next neurotransmitter, and it affects mood, hunger, sleep, and arousal. And arousal means like if you're awake or not, if you're feeling sleepy, if you fall asleep in class, that means your serotonin levels are probably decreasing. If you're really upset or if you're in a bad mood, you probably have an excessive amount of serotonin affecting a certain part of your amygdala, which we'll talk about next unit. So not only do different neurotransmitters do different things, they also affect different parts of the brain, as you can see in this illustration, between the serotonin and dopamine pathways. This further explains the different effects of serotonin and dopamine. So neural communication refers to how the neurons relay messages to each other. It is referred to as electrochemical reaction. Neurons are surrounded by fluid. The fluid inside a neuron contains negatively charged atoms called ions, from the fluid located outside the neuron, which has positively charged ions. The neural membrane only allows certain ions through the membrane. positively charged ions, sodium and potassium ions, and negatively charged chloride ions flow back and forth across the cell membrane, but they do not cross at the same time. The difference in the flow leads to a higher concentration of negatively charged ions inside the axon. So here's a diagram of everything we just explained. All right, so pretend that this wall here is the wall of the axon, and this is the inside of the axon, and this is the outside of the axon. Okay, and remember the axon being the long branch of the neuron. And outside the axon, we have positively charged potassium and sodium ions. And on the inside of the axon, we have negatively charged chloride and protein ions. All right, so what happens is when action potential takes place, the ionic chamber opens up, releasing the negatively charged ions outside and the positively charged ions inside. What this does is it depolarizes the inside of the axon, giving it a spark, an electrical charge, which will then accelerate action potential further down the axon. So there are three phases of communication within a neuron. All right, action potential, the neural impulse created when a neuron fires. This is the impulse that travels from the dendrites down the axon to the terminal branches. Well, after action potential takes place, it goes through what's called a refractory period. Okay? The brief instant, this is a very, very brief instance when a new action potential cannot be generated because the neuron is recharging or reloading, if you will. All right, so it's sending all those neurons back into the neurons, it's sending all those neurotransmitters back into the neuron from which it came so it can charge, so it can fire again. All right, when a neuron does nothing, when a neuron is just sitting there with potential to be fired, this is called resting potential. So positive ions will flow into the neuron if not stopped or pumped out by the membrane. This is called the electrical potential, which is measured in millivolts. The resulting potential is the neuron's usual charge, which is about 70 millivolts. When the resting potential has changed enough, about 10 millivolts, the membrane changes, and this is called the action potential. So action potential, this is the neural impulse. It's a brief electrical charge that travels down an axon, each tripping the next. This is known as depolarization. Generated by the movement of positively charged ions, also known as atoms, in and out of channels of the axon's membrane. The speed at which an action potential travels the axon range from 2 to 250 miles per hour. So here we see the axon. All right, here's the axon membrane or axon wall. Here's the outside of the axon, and then everything in the middle is the inside, okay? And so here we see the positively charged ions flowing into the center of the axon, which depolarizes or creates an electric charge, if you will. This spark will then pass on to the next ionic chamber, which then will open up, allowing the positively charged ions to flood in, creating another spark, and then passing on to the next, and to the next, and so forth. All right. So depolarization, again, is when the electrical charge of the cells moves towards zero. Polarization is when the electrical charges of a cell moves away from zero, as we see here. They're moving away from zero, whereas in depolarization they move towards zero. Okay, so again, the refractory period is a resting pause when the neuron pumps the positively charged ions back outside, then it can fire again. Excitatory is when the signal to send is a signal to send the message. All right, it's a signal for the neuron to fire. An inhibitory message is a signal for the neuron to stop firing. All right, the threshold, like we said, is the level of stimulation required to trigger a neural impulse, and this takes place in the nucleus of the soma. An agonist is a drug, okay? It's something that we take, like a medication, and an agonist excites by mimicking particular neurotransmitters or blocking their reuptake. Antagonists inhibit a neurotransmitter's release or block its release. All right? This is really important for understanding how drugs affect us, so make sure you know the agonist and antagonists. So here we see agonists. They are mimicking the natural neurotransmitter that we have in our body. Maybe we don't have enough of those neurotransmitters, so we need to take an agonist medication to make up for those neurotransmitters we lack. All right? Or maybe we have too much of a neurotransmitter, so we would take an antagonist, which would block the neurotransmitter from being accepted into the dendrite, and it pushes it back into the end bulb from which it came. This shows how quickly action potential takes place. So here we see the resting potential, the neuron is ready to fire, and so when the stimulus's threshold is surpassed in the nucleus of the soma, then action potential takes place within less than a second, and then the refractory period resets the neuron, leading to resting potential. So again, here we see the end bulb, we see the vesicles containing the neurotransmitters. When action potential reaches the end bulb, the vesicles pop, releasing the neurotransmitters, crossing the synapse, and into the dendrite of the adjacent neuron. Also, neurons fire or they do not fire. This is called the all or none principle. A neuron can't halfway fire. It's like a gun. You either shoot a gun or you don't shoot a gun. That's the way it is for a neuron. The neuron's reaction is an all or none response. So why don't you go ahead and pause the video for a second, read through this slide, analyze it to fully understand the depolarization of the axon during action potential. You should be able to explain this chart by now. All right, see you guys in class.